All right, guys, so we're going to go over our exam review, and if you complete this with me during the video and turn it in, you will earn some extra credit points on your test. So first of all, number 1 says make sure you can identify the parts of the cell membrane. This is in your notes, your cell membrane notes. You should be able to identify what a phospholipid is, what the hydrophobic and hydrophilic ends are, which we will do in one of these questions, that there is a phospholipid bilayer, and what a channel and a carrier protein look like. All right, so let's discuss the functions of the cell membrane. So the main function of the cell membrane is to control what enters or leaves your cell. Okay. It also maintains something called homeostasis, which is keeping a constant internal environment. And it is also a barrier between the inside of the cell and the outside environment. Okay. Describe the structure of the cell membrane. So it is made out of a phospholipid bilayer for 2 layers, and some proteins. And there's lots of different proteins, but the 2 we're focusing on are the channel proteins or carrier proteins. Okay. This says draw a phospholipid and label it. So I'm going to insert some parts here. I'm going to get a little circle. Let's put that guy right there. And then let's get a little line. I'll put that right there. And then we'll grab another line and put that on this side. Oh, that didn't work. Whoa! Okay, that's all right. We'll drag it over there. All right, so that's kind of what my phospholipid looks like. And it wants me to identify the hydrophobic and hydrophilic parts. So we're going to insert a text box, just a simple text box, and it comes out huge. But then we're going to make this hydrophobic. The hydrophobic part of your phospholipid is the fatty acid tails because they're made out of fat, and fat and water does not mix. I'm going to copy this and paste it, and paste it. There we go. And we'll make this one hydrophilic. That means it likes the water. And it's the phosphate heads that like the water. All right, so there we go, hydrophobic and hydrophilic. What is needed to transport very large molecules that are insoluble in lipids across the cell membrane? That's going to be your proteins. All right, your proteins help to move large things across. There we go. All right, down to cell transport, make sure you can identify diagrams of endocytosis and exocytosis. So those are in your notes, as well as we drew them on the homeostasis and the cell membrane worksheet. Diagrams associated with hypertonic, hypotonic, isotonic. So we did the interactive notebook with that, and also the beaker worksheet. So it says, what is the difference between active and passive transport? So in passive transport, you're going to be moving from high to low concentration, and there is no energy needed to do that. And so the opposite is that in active, you're going to be moving from low to high concentration, so against the gradient, and it's going to need energy. It needs energy. So passive with the gradient and active against the gradient, which goes with the next 2 questions. Active transport does move against the gradient from low to high, so remember, think about my little mountain there, so we'll highlight that one. Passive transport moves with the gradient from high to low. Number 5 lists 3 types of passive transport and why they're different. So let's start with diffusion. All right, and they're all passive. So they're all high to low. They're all no energy. The difference is this is the movement of small particles, things like oxygen and carbon dioxide. Then we have facilitated diffusion, which is still diffusion, so still high to low, no energy, but this is movement of large molecules, all right, sometimes charged molecules. And then we got osmosis. all right, which is still also, still going from high to low, still does not need energy. That's what defines it as passive. But also, this is the movement of water. Okay, so this is what differentiates them, what's being moved. But they're all going from high to low, all no energy. What kind of transport does the sodium-potassium pump complete? Well, all pumps need energy, so this is active transport. And what comes in and what comes out? So sodium, which is going to be Na, goes out. You want to get rid of the salt. That's part of salt. And potassium, which is a K, weirdly, goes in. You want to keep the potassium. That's like from bananas, good for you, and get rid of the sodium when you're talking about the sodium-potassium pump. What kind of transport is endo and exocytosis? So these guys are when you're making that vesicle bubble and moving it around. It takes energy to do that. So these two are active transport. All right. Oop, I wrote active twice. So you also can know it's active transport if it's not one of the three kinds of passive. So active transport, and then we can say endocytosis brings in materials using a vesicle. Okay. Exocytosis, exo for exit, right? Brings materials out of the cell using a vesicle. Remember, those vesicles are just specialized vacuoles. Compare pino and phagocytosis. So these two things are both types of endocytosis. So phagocytosis, think of phagocytosis like the cell eating, but it's bringing in large things that's eating, okay? So phagocytosis brings in large things. They're things like bacteria, using a vesicle, because it is endocytosis. Whereas pinocytosis is more like drinking. This is when the cell brings in small things like water molecules or anything dissolved in the water molecules, brings in small things, like think small like the head of a pin, using a vesicle. There we go. On to the next page. What would happen to an animal cell put in a strong sugar or salt? Doesn't matter if it's sugar or salt because they're all solutes. If there's strong sugar or salt, that means it's going to be hypertonic, and water always moves towards the hypertonic. So that means water is going to leave this cell. So an animal cell would basically shrink or shrivel. What would happen to a plant cell put in the same situation? Well, the whole plant cell doesn't shrink, just the insides, because that cell wall is rigid and protects. So this is something called plasmolysis, all right, where you break the plasma. So you just shrink the inside. How would a plant or animal cell react to be put in distilled water? That's pure water, so that's about as hypotonic as you can get, which means your cell is hyper and the water is going to go into your cell. So an animal cell would pop. It would swell and then pop, right? Plant cells are not going to pop. Instead, plant cells will experience something called turgor pressure. All right, that's when the vacuole fills up and pushes against that cell wall. What is it called when the concentration outside the cell is equal to in cell? That's isotonic when they're the same. All right. It could also be dynamic equilibrium. Okay. But on your test, I'm going to be looking for probably isotonic. All right. Then we're on to cell division. Types of cells produced. Well, mitosis, like mitose, those are somatic cells, which are body cells. And meiosis, remember, me is sexy. So that's your gametes. Gametes are your sex cells. In mitosis, you divide once. In meiosis, you divide twice. If you only divide once, you get 2 daughter cells. And if you divide twice, you get twice as many, so you get 4. If you're making body cells, you want them to have a full set of chromosomes, which is diploid, di, because you have 2 of each kind. In meiosis, your sex cells are half, because you take half in the sperm and half in the egg and put them together, and you get a whole again, right, with a baby. So those are haploid. Diploid is 2N, because you have 2 of each kind of chromosome, whereas haploid is just N, because you have half of that. When you're making body cells, these are going to be identical to the parent cells. If you're replacing your skin, you want it to be the same. Whereas when you're looking at your sex cells, they are genetically unique. And that's why brothers and sisters look so differently, because you can mess your DNA all up and get a lot of variety there. In a normal human, how many chromosomes are present? And our diploid number is 46. So our haploid number is half that, that's 23. And there's nothing sexual about making skin cells. Oops, so I wrote that. So that is asexual. And then, of course, making your sperm and egg is sexual. All right, down below, be able to identify the parts of a chromosome. It says draw a chromosome, label chromatid, and a centromere. So again, I'm going to go to Insert, Shapes, and I need like an X shape basically. So there's one right down here in equations. So I'm going to take that and use that as my chromosome. And then I'm going to insert a text box again. And I'm going to name chromatid. So chromatid is essentially one side of my chromosome here. Okay. And then my centromere. Copy that guy, and I'll say is center, right? Centromere center. Make it a little longer. All right, that's going to be right in the center. And so you could put lines in there if you want, but for the sake of time, because this can only be so long, I'm moving on. All right, what are homologous chromosomes? They are the same size, same shape, same traits. Okay. Somatic cells are body cells. Why is that blue? I don't know. Gametes are sex cells. All right, name of the parts of the cell cycle in order. So notice this says cell cycle, the whole thing. So we have interphase, which consists of G1, S, G2. Then we go into mitosis. Let me put a comma there. Or meiosis, but in the case of mitosis, we have PMAT as the 4 in order, and then we have cytokinesis. Okay. What is the longest phase? Well, ABC is the longest phase there. The longest phase is interphase. That's when your cell is not dividing. But it is preparing to divide. Cell singular. Okay. What occurs in G1S and G2? That's going to be in G1 we have growth. Then in S we got replication. That's when you copy your DNA. And then in G2 we have preparation. That's when you're making everything else. All right, compare and contrast cytokinesis in plants versus animal cells. So in plants, you make a cell plate, and in animal cells, you make a cleavage furrow. So we got quite a little bit left, so we're going to run out of time with this video. I'm going to stop it and restart at number 10.